This is a production of Cornell University.
So thanks for being here. Good afternoon, everyone. Myself, Srikanth. I'm a second-year PhD grad student in plant breeding and genetics. I'm from Mueller lab. Today, I would like to explain the project that I'm working on from past couple of months. And before diving into the project, I would like to explain some of the fundamental concepts and essential to build this project. So first, what exactly is a database? So database is the system that is widely used across the industry and academia for data storage, data retrieval, and data management. So BreedBase. So BreedBase is the relational database system that is free and open source that our lab developers have been built over the years. They added a bunch of features based on the plant breeding community's request. And now plant breeding communities, they can easily upload their data into the BreedBase, and using the analysis tools that our lab developers have built, they can analyze their data and make data-driven decisions. So using the breed-based instance, our lab also involved in developing a couple of crop databases, specifically for the cassava base, Musa, that's for banana, crop for base, and sweet potato base. And there are many other crop breed-based instances used by companies, but I cannot list them. With this, even in the past year, I worked on repetitive measurements. In other words, you can say this as a time series feature. It's basically where you have a multiple values for a given trait that you would like to store. So this feature was requested from UC Davis breeding program and North Carolina State University breeding program. Therefore, we've implemented that. So with this, now I have a very good understanding of how breed-based works and how the process of breed-based actually is implemented. So with this knowledge, knowledge and experience, I would like to take breed-based to the next level, where I would like to integrate artificial intelligence in the breed-base. So before actually diving into the what AI application we will be building for the breed-base, I would like to give the brief statistics about the cassava base. As I mentioned in the previous slide, cassava base is one of the largest database as we have so far. So as of March 2025, we have almost 240 GB of data in the cassava base, out of which, out of which 12 GB is phenotypic data, and why it is not working? Okay, yeah, 118 GB is of genotypic data. And due to time constraint, I'll not go to each component of this. I'm not sure why this is not working. Okay, yeah. Due to time constraint, I cannot go through each component. However, I would like to pinpoint and mention that cassava base has been widely used across the world in almost 550 locations. As you can see, this database is so much complex and it's a large database, right? So we often use a structured query language, which is called SQL language, to communicate with the databases. This is not the observable trait in most of the plant breeders or the community, right, to communicate with the database. With this in mind, we would like to build we'd like to develop a chatbot for the breed base using LLMs. So first, I would like to show you the backend or the workflow, how it looks after it has been implemented or deployed in the production site. So imagine you are a user and you are communicating with some AI. I hope everyone has used ChatGPT or Google Bard, so they know, right? So user might ask, hey, how many trials do I have so far in IITA breeding program? So then using the RAG technique that I'll describe in the next slide, they will extract the trial column names and the table names from the database for this specific NL question that user asked in the interface or in the chatbot. So these both will be provided to the fine-tuned LLM, and LLM will write SQL query, which means it's another word where you use to communicate with the databases. So this will eventually be executed on the database, and that's where you get the output. So we do not like, in addition to showing the value from that we retrieved from the database, we would also like to show the generalized statement along with the value. Therefore, this would be clear and concise information to the user, whoever is querying, right? Yes, of course, it is true that there are so many closed source LLMs that are outperforming in various tasks, such as summarization, code generation, and a lot many other tasks. However, recently, Stanford NLP group have tested with the various closed source models. They fine-tuned their LLMs using those closed source API keys, and they found that there is an ineffectiveness in updating existing knowledge. So what I mean by that is, once they fine-tuned their models using these closed source APIs, after that, they modified their data in the database. And fine-tuned LLM couldn't able to retrieve the updated data. And there are a wide variety of reasons why this is happening. But the primary reason is closed source LLMs doesn't have a flexibility of using different LLM techniques on the fine-tuned model. Therefore, you cannot even improve the model accuracy or performance, right? So with this, I referred a couple of times about the closed source LLMs. So primarily, those are developed from OpenAI and the Google and the Cloud Anthropic. And the open source LLMs are Meta, DeepSeek, and Microsoft. These are primarily deployed on the Hugging Face. From Hugging Face, you can easily deploy onto your local machine or the GPUs, and then you can run them. So for this project, we will not be using these closed source models. However, we would be likely to use open source LLMs to build this project. So I mentioned LLM strategies in the previous slide a couple of times, right? First, I would like to mention about retrieval augment generation. So as it is a $5 word and it's a simple word, as the name it indicates, it's just a retrieving. So basically, if user asks some question in the NL interface, it retrieves the table names or the column names with respect to the database to that question. To that question. So it's not a high level of coding that is required to implement this technique for this objective. And next comes the supervised fine-tuning. Supervised fine-tuning is a time-consuming and it's an iterative process. And it's you need to build a training data set in a way that it should include all the edge cases even. So it's a high level of coding that is also required. So last technique is the reinforcement learning. So reinforcement learning is a branch of ML that we actually basically use the AI, train the AI agents based on the trial and error method. And it's a high level of coding that is required. There is no straightforward mathematical framework or a mathematical formula to directly deploy them. You have to verify with the different algorithms for this. With that, I would like to thank my PI for giving me exciting opportunity to work on this. And also, I would like to thank John for giving me access to the GPUs to test it before trying to deploy these models on the production side. With that, I would like to open the floor to any questions. Thank you so much. Yeah.
Last slide, you listed a couple different strategies. Is there any one that you want to look at going forward, or are you going to try all three maybe to see which one works best?
Yeah. So the question is, in this slide that I'm showing right now, she mentioned that I showed here different techniques, right? Do I use all of those, or do I use only one among the three? So these three techniques are very important in order to make this objective efficient. So you cannot use only one. So these three techniques are combined to build this chatbot, meaning all these three techniques are very useful. So it's doing one technique is doing one job. You have to combine all these three techniques. Yeah.
So you're using the cassava data set to do this. How hard would it be to then use another data set for another crop, like to translate this to another crop? Would you have to rebuild and train the model all over again, or would it be easily transferable for different crops?
Yeah. The question here in the audience is, currently I'm using the cassava base for the initial stage to build this. If we eventually in the future, if I transfer to the other crop or other databases, how easy or how hard is it? So the simple question is, for our example, we have a multiple crop databases, right? So for cassava base and most of the databases, we have a same structure. So what I mean by structure, so the table names, column names, all are same across the different crops. Only the values or some functionality might change, right? So if we implement cassava base and it's working fine as we expect, so in order to scale up to the other databases, we need to change the table names and the column names in those databases to specifically to those crops in order to make it. Yeah, that's it.
So my name is Monserrate. I'm a four-year PhD student in Larry's lab in Cornell Agritech in Geneva. And today I would like to do an updated summary of my work on mapping QTL and identify candidate genes for seed and agronomic traits in hemp F2 micropopulations. So to start off, I would like to, for some of you that are not familiar with this crop, there are three main market classes in hemp: flower, fiber, and grain. For the flower, obviously the end product is the extraction of cannabinoids for these flowers. But for all these market classes, there are key traits to take into account when considering cultivars and when breeding cultivars. For the flower per se, the idea is to increase the floral biomass. So flowering time is one important trait to consider, and obviously cannabinoids as well, which is not something that I'm going to cover today. For the fiber, the idea is to harvest the stem. So we also want, based on the idiotype you can see here, for example, is a tall, non-branchy, late flowering cultivar. So one of the important traits is flowering time as well as biomass to ensure that we have this accumulation of biomass, and fiber quality is another important trait. So from this biomass that is accumulated, we can remove this bast from the herd for the to maximize the bast production for textiles or the hair production if the intent is for construction materials. And lastly, we have the grain. Another market class that is embedded in it is the dual purpose, which stands for production of the grain and the stalk. So you harvest the inflorescence and then you harvest the stalks at the end, so you can get fiber and grain as well. And here, again, one of the key traits to consider is flowering time, because we want to ensure that we accumulate biomass so this crop reaches a sufficient height for an optimal harvest, and meets the goals, whether it's dual purpose or grain only. And biomass as well, because we want to have that balance between certain accumulation biomass for both fiber and grain. And obviously, I'm not gonna forget grain yield and seed quality, especially seed quality is another important trait. In this case, we are considering quality right now, is the seed size as well as the protein oil content. But speaking of seed size, so here in this picture, you can see the variation on seed size that we have in our germplasm in Geneva. And from an importance agronomic standpoint, actually, seed size could be beneficial for stand establishment. From a consumer perspective, a larger seed will be more appealing for a consumer. And from a processing point of view, we have observed that larger seeds may lead to a better processing of these seeds. So traditionally, hemp hearts that you see and you can access commercially are seeds that originate from cultivars that are not that different from seed size that equates Białobrzeskie or Felina 32 that you see here, or perhaps even smaller than this. So we're interested in looking at the processability of these larger seeds that you see on the left, as well as, of course, the genetic mechanisms underlying that trait or the seed size alone. So this hemp grain, which is not grain, it's an akin actually, once it's harvested, it is processed, and here you can get two main components of these seeds, the hemp hearts, which are actually the cotyledons of the seeds, and the hemp hulls. The hemp hearts are high in protein, about 20 to 25% protein. The fat is about 25 to 35, if I'm not mistaken. And the hulls, which are the pericarp of this akin, is high in dietary fiber, about 25 to 35% as well. But ultimately, the end product of these components become into hemp seed meal, hemp seed oil, fortified foods like baked goods, as well as plant-based meats and dairies, as you can see here highlighted in this graph. So with all of this in mind, I established three objectives, and those are to identify significant QTL for seed size and agronomic traits, and to compare or determine the consistency of this QTL between three populations that will represent each of the market classes, and identify, hopefully, the candidate genes within the 1.5 log interval for each of these QTL. So to start off, in 2021, a high cannabinoid F2 population was developed, crossing a paramildew-resistant clonal individual F58 and TJCBD clonal individual that is paramildew susceptible. The idea here, or the main goal of this was to characterize paramildew resistance, as I was saying. However, other phenotypes were measured. For this, the crossing scheme was the selfing of two selected F1s, generating two F2s. And the idea of having more than one F2 is so that, because hemp is highly heterozygous, having more than one F2 population increases our likelihood to capture the effect of the allele present in the parents. So here we have 21,004 and 21,005 with 366 and 304 individuals respectively. Another important thing to mention is that the flowering time between these two parents is about a week. So they flower relatively, I wouldn't say early, but not too late either. For the grain population, in this case, we employ a dual-purpose breeding line. This one is 2011-79, or GBAH 2011-79, which is a late flower individual, large-seeded, and crossed with a piccolo, which is a very popular Canadian grain cultivar that has high protein content, that has small seed and early flowering. And in this case, the crossing scheme was developed to a seed mating of the F1s, and we ended up phenotyping only the females. 198 of them. For the fiber, in this case, we use a fiber breeding line to GBAH 191024. This is a very late flowering, a very large seeded cultivar population. It was crossed with the first New York feral population isolated, which is Logan. This is a very early and a very small seeded cultivar. In this case, we also did seed mating for the generation of the F2s, but here we phenotype and genotype females and males, having a total of 445 individuals. For the creation of the linkage maps, we use two genotyping platforms, sorry. For a high cannabinoid population, 21,004 and 21,005, we use an Illumina array developed by Phylos. Here we ended up with linkage maps that have 2,659 and 2,197 markers respectively. Whereas for the grain F5 population, we use an Agilent Sure Select prop set that we developed in our lab, which we named Can Select. This eventually helped us develop or create a linkage maps that have 748 markers for the grain and 922 markers for the fiber. So with all this in mind, on all these methodologies, so I decided to look at the seed size. In this case, we quantified that by the weight of 1,000 seeds, or 1,000 seed weight, or TSW, as you can see here. First, in the high cannabinoid population, 21,004, we found 5 QTL that are associated with this trait. And as you can see here, the largest peak is located in chromosome 1, that explains a good portion of the variants, and it's a very small region, relatively speaking, in the genome. And we see that the effect of the A allele is dominant over the B allele. For the A population, we also find a QTL in chromosome 1, but conversely, this is the only QTL that we find explaining low proportion of the variants, but in a very large region. And in this case, the effect of the A allele over the B allele seems to be additive. For the grain, we have 4 QTL identified, and in this case, we don't observe this. We observe that the largest peak is in chromosome 9 instead, explaining a considerable part of percentage of the variance, sorry, but a large region, and also seems to be additive. And in the fiber, interestingly, we only find 2 QTL, but a massive peak in chromosome 4 that explains over 60% of the variance in our population. But unfortunately, the region is quite large, it's 9.1 megabases. And here, the additive effect, the effect of the B allele over the A allele is additive. Now, for the flowering time, in this case, for 21,004 in the high cannabinoid population, we find one large peak in chromosome 1 again compared to the 1,000 seed weight. That explains also a good proportion of the variance, and it's a small region. The effect of this allele is again dominant compared to the one identified in the 1,000 seed weight. For 21,005, we also observed another peak, well, 2 peaks actually, but the largest peak in chromosome 1, explaining 20% of the variance in over 20% in a large region. And in this case, the effect is additive. For the grain, we only find 1 peak in chromosome 1 that explains over 0.5% of the variance, and in this case, this allele seems to be additive. And for the fiber, we observed the most QT identified for flowering time, 4, 1, 2, 5 actually, to be precise. But again, the largest peak is in chromosome 1 with an additive effect, but unfortunately doesn't explain that much of the variance. So what you see here highlights the importance of this chromosome 1, or the presence of flowering time genes in this chromosome 1 in the cannabis genome, at least for genomes that we're evaluating here. And lastly, for biomass, which I would expect that will coincide with the flowering time, and for this family, it does, explaining proportion of the variance, same with the genetic region. But again, this is an additive allele compared to the B allele. For high cannabinoid 2005, again, there's a peak here. There are not many that doesn't explain much of the variance, but it's still there. And I'm seeing the same thing in grain. The largest peak, or the only peak in chromosome 1, equates to the one in the flowering time, explaining good portion of the variance, but a large region. But in the fiber, however, we don't observe that peak in chromosome 1 that should coincide with flowering time, but instead it's only in chromosome 6, which is very interesting for us. However, it doesn't explain that much of the variance, but is interesting nonetheless. And yeah, so with that, the key takeaways are that the chromosomal location for largest effect QTL for seed size measured as 1,000 seed weight differs depending on the location on the population that we use. The largest effect of this QTL for flowering time in all population was located in chromosome 1. The largest effect QTL for biomass seems to coincide with that for flowering in all populations except the one in fiber, which is in chromosome 6. And one thing, I don't know if you caught it, but there seems to be a one pleiotropic dominant effect QTL in chromosome 1 for this high cannabinoid population in 21,004. So with that, I would like to thank all the members of the Larry Smart Lab, as well as my funding sources listed here, and I'll welcome any questions.
Big range of planting densities for hemp, like super dense for fiber, super sparse for cannabinoids. How'd you pick the density or the densities used for this study? And does that have like a complex, does that impact flowering time and total seed weight differently, like in the fiber biparentals versus like the cannabinoid or grain biparentals?
Yeah, so the question is how I decided the planting density and if that affects the traits phenotype here. So the planting density was Y spacing. So for example, one trait that will ultimately be affected by a planting density is yield for biomass as well as seed yield. That definitely affects. Flowering time, however, does not seem to be affected by the planting density, and the seed size does not seem to be affected either as well. Emily.
So I was wondering, you have a lot of QTL on chromosome 1, and
Yep.
you said it might be due to the pleiotropic effects from that one parent population. Have you looked further into those QTL to see if it's like one or two major genes that seem to be controlling this effect, or are the QTL in like very different locations on that chromosome?
This is why I like you, Emily. Okay, so I did not have the time to incorporate this in the presentation, so I was hoping somebody would ask that. So we do have the sequencing, the actual sequence of the parents in this high cannabinoid population, and we were able to have, what is that called? The structural and functional predictions of the genes located. So what I was able is to section the regions of that specific QTL and compare them with each other and see what differs. And the one that I identify as the one differing is this LUK7-like protein in chromosome 1. And there is this G insertion that causes a frame shift mutation in this haplotype. This LUK7-like protein is, it's homolog to a LUK7RL in the Arabidopsis, and this seems to participate as an accessory protein to the U1 small nuclear ribonucleoprotein in the spliceosome. So it's affected or affects the RNA maturation of several genes. And a group in Germany actually validated this by knocking it out and testing like what is upregulated or downregulated and what several outcomes that they got from it, in which it highlights a pleiotropic effect in the Arabidopsis organism.
This has been a production of Cornell University, on the web at cornell.edu.